All right, let's do something amazing. Today, we're going to take a journey, a tour, right to the center of the eukaryotic cell. This is the tiny, microscopic city that is the foundation for pretty much all the life you can see with your own eyes. And think about that for a second. From the biggest tree you can imagine to the tiniest bug, from you to me, we're all built from this exact same incredible blueprint. So if we can understand this blueprint, we're really understanding the engine of complex life itself. Okay, so when you look at life on Earth, there's this huge fundamental split. On the left, you've got the simple, smaller guys, the prokaryotes. Think bacteria. And on the right, you have us, the eukaryotes. You can just see the difference, right? They're bigger and they're full of all this internal structure, all those little compartments. Those are called organelles. So come on, let's step inside and have a look around. And the name itself, eukaryote, actually gives you the biggest clue. Eu means true, and karyon means nucleus. So this is the cell with a true nucleus. That's the main event, a special protected room just for the cell's genetic material, its DNA. This, plus all those other organelles, creates an amazing division of labor, where every part has its own specialized job to do. Every good city tour starts at the border, right? And for the cell, that's the plasma membrane. But I want you to forget any idea of this being a static wall. This is a smart, dynamic gatekeeper that controls everything that happens between the inside of the cell and the great outdoors. So what you're looking at here is what scientists call the fluid mosaic model. It's a fantastic name. Fluid because that main structure, that double layer of lipids, is constantly shifting and moving, which lets the cell be flexible. And mosaic because you have all these different proteins embedded in it, like tiles. And those proteins are everything. They're the gatekeepers, the sensors, the doorways. They make this barrier incredibly selective. All right, we're in. But wait a second. If the border's fluid, what stops the whole thing from just collapsing into a puddle? Well, that is the job of the cytoskeleton, the cell's incredible internal scaffolding. And this thing is an absolute engineering marvel. It's basically made of 3 different types of protein fibers. You've got your microfilaments. Think of those as the cell's muscles. Then you have these rigid, hollow microtubules. Those are the bones, the steel girders holding everything up. And finally, you have intermediate filaments, which are like these unbelievably strong ropes that give it tensile strength. This whole internal network is what allows eukaryotic cells to have all these complex shapes and even to move. And just look at the specs on these materials. It's wild. The microtubules are literally millions of times stiffer than the intermediate filaments. But then, check out how far they can stretch. Those ropes, the intermediate filaments, can stretch to more than double their length before they even think about snapping. The others are much more brittle. So you get this perfect combination of rigid bones for support and super stretchy ropes for toughness. It's brilliant. But here's the coolest part. This isn't just some dumb static scaffold. It's a smart material. The more you push on it, the stiffer it gets. It's a property called strain stiffening, and it's basically a built-in defense mechanism. It helps stop the cell from getting ripped apart when things get rough. Okay, let's head deeper into the city, right into the downtown core where all the action is. We're going to check out the organelles, the specialized factories and departments that make this whole operation run. And here it is, the big cheese, the star of the eukaryotic show, the nucleus. This is the cell's library, its city hall, its command center, all rolled into one. It's wrapped in a protective double membrane, and you see all those little holes? Those are nuclear pores. They're the secure channels that let information pass in and out. Now get this. If you took all the DNA packed inside one single human nucleus and stretched it all out, end to end, it would be about 2 meters long. 2 meters! So how on earth do you cram that into a space that's just a few microns wide? Through this insane, beautiful process of folding and coiling it around proteins to create what we call chromatin. It is a masterclass in data compression. So you've got the blueprints. Now what? Well, the cell has this incredibly efficient assembly line. It all starts in the nucleus, where the DNA instructions are read. That message gets sent out to the endoplasmic reticulum. That's the factory floor where the proteins are actually built. From there, they head over to the Golgi complex. Think of that as the shipping and receiving department. It sorts, packages, and puts a shipping label on everything. And finally, the finished proteins are shipped out in these little bubbles called vesicles to wherever they need to go. Right, but a factory this busy needs power. A lot of power. And that's where this guy comes in, the mitochondrion. You've probably heard it called the powerhouse of the cell, and that is exactly what it is. This is the organelle that generates ATP, the energy currency that pays for literally everything the cell does. And see all those crazy folds on the inside? That's all about maximizing surface area to crank out as much energy as possible. Okay, but here's where the story takes a really weird turn. Mitochondria, they have their own DNA, and it's circular. They also have their own ribosomes, the little machines that build proteins. And both of these things are totally different from the DNA and ribosomes in the rest of the cell. Now, why would that be? Just tuck that little fact away for a second. It's the key to our final, biggest question. So we've had the grand tour. We've seen all this incredible machinery, this organized city. But that just leads to a much, much deeper question. Where in the world did all this complexity even come from? I mean, really, how did eukaryotes come into existence? This has been one of the biggest puzzles in all of biology. How do you make the leap from a simple little prokaryotic bag to this bustling, compartmentalized city of a cell? Well, the leading explanation is as elegant as it is mind-blowing. It's called the endosymbiont theory. And the basic idea is that this complex cell isn't really one organism at all. It's the result of an ancient merger, a partnership between separate, simple cells. So here's how it probably went down. Way, way back, maybe 1.5 billion years ago, you have a large primitive host cell, and it engulfs a smaller bacterium. But, and this is the crucial part, instead of digesting it, they form a partnership. See, that little bacterium was really good at using oxygen to make energy. So the bacterium got a safe home, and the host cell got a massive, reliable energy surplus. And that little bacterium, over eons, eventually became the mitochondrion. And the proof for this wild idea? It's sitting right there inside the clues we found earlier. Remember that weird little fact? The DNA in mitochondria is circular, just like in bacteria. Their ribosomes are a bacterial-style ribosomes, completely different from the ones in the rest of the cell. The exact same evidence exists for chloroplasts in plant cells. It's a living fossil record of a partnership that completely changed the course of life on Earth. So, eukaryotes are these incredible marvels of complexity and efficiency, no doubt about it. But then you think about the prokaryotes. In all their simplicity, they have dominated this planet for nearly 4 billion years, surviving every catastrophe imaginable. It kind of leaves you with a final thought, doesn't it? What does it truly mean to be successful in the grand story of life? Is it complexity, or is it sheer unyielding persistence?